I think we're ready to go, baby. Let's get this bread.
My reading aids on.
Yeah.
All right, I am thoroughly stoked to be introducing Matt Nicholson on the occasion of his PhD defense. Matt is a local boy, and he was born in Miami Beach and grew up in Pembroke Pines right up the road. And so he grew up idolizing the U. So this is in many ways a dream come true for him. He did his undergraduate work at FGCU across the peninsula. And then after that, he went to South Africa and worked on some shark projects there. And then came back from South Africa and worked serving alcohol to land sharks in South Florida. Yes.
Yeah.
So heard the Jimmy Buffett song, Sharks. That's kind of was his gig. And then he went off to went to grad school at University of Exeter, where he graduated with distinction in 2014, working on seabird trophic ecology using stable isotope analyses, which were fairly new at the time. Then he realized that there was something more exciting and charismatic than sharks, and that was parasites. And so he joined us as a volunteer field assistant. And so like we do with most of our volunteers, we explain to them that this is not easy work. It may look exciting, but it's a lot of work and a lot of long hours, and we stay up late with the musicians and really want to do this. And so not only did he do it and thrive, but he pushed the envelope. Like, he made it go to another level of insanity. And so it's like, okay, man, this guy's for real. So he joined us as a PhD student then in 2016 at Arkansas State. And so things were humming along quite smoothly for a few years, maybe actually a year. And then hurricanes Irma and Maria decided to visit our main study site and destroyed everything. And Matt was supposed to join us in South Africa for a meeting to present some of his work there, and he emailed us and said, sorry, but like, this place is trashed and I can't get out of here, and so you have to go without me. So that completely destroyed everything. It's lost samples, lost artificial reefs, like everything we had there was gone. And so it took really about a year to recover from that. And then, of course, by the time he recovered from that and was catching up, he got hit with COVID. And that set us back, or him back, everybody back, really. So he had to endure usually within a PhD, you have to endure sort of one crisis. He had to endure two, basically back to back. And still, so given that he's finishing now is really impressive, actually, given what he's had to endure. So as working with anybody in the field for 7-8 years, you've got a lot of stories to tell, so I don't have time for them. Plus, most of them fall under the category of what happens in the field stays in the field, so I can't talk about them. But there are a few that come to mind. One of them was when he was, I can't remember whether it was when you were in your volunteer days or early PhD student days, but we had to transport a bunch of stuff from this remote field station in St. John back to St. Thomas and store it in our facility there. And I was convinced that there's no way we're going to get all the gear in this one vehicle. I was always making plans to go over, drop it off, and come back again for another load. And it's like, okay, let's just go deal with it. And Matt goes, no, we can do this. I can get it all in here. It's like, no way. And we did. We got everything in there. And so that was impressive. And another one that came to mind was that one of the few places we work, Guana Island is a private island in the British Virgin Islands, and most people go there for vacation. But we were there working. I wasn't there, but Matt was running the team. And I got an email from the dean of the Yale School of Medicine saying, I met your PhD student there, and he was running the team there. And I just can't say enough about how impressed I was with how he ran things and how hard that team worked. And I'm going to use that as inspiration to discuss with my postdocs and my PhD students. It's like, okay, great, way to represent. Perhaps the thing that really has been the biggest benefit to us as a team is that Matt is a natural stand-up comedian. If he were not doing this, he'd probably be doing stand-up comedy. And so in the field, things can get kind of rough. Having somebody who's always keeping people laughing is just like really wonderful to have. And so, yeah, obviously I can go on and on, but I won't, and I will let Matt have the floor.
Cool. Thanks, P. I did not prepare a stand-up routine today, so thanks, Paul, and thank you everybody who showed up today and everybody who's here on Zoom. Before I really get rolling, I wanted to share this quote and also share this picture. This quote really sort of summarizes what drives me in research, which is just the fact that there's no feeling like discovery, like answering questions for the first time, which is what I always say is just the thing that makes research the only thing I could do is answering questions that have never been answered before and discovering the knowledge of tomorrow. And one of the first times that really hit me was in this picture. This picture is from 2010 when I was at Florida Gulf Coast University, and we were collaborating with the University of Miami. We were collaborating with the shark team. And while the shark team was doing satellite tags, we were taking muscle samples for stable isotope analysis and stuff like that. And growing up as a kid who was a huge fan of movies like Jaws and always was just sort of enamored with the ocean, and growing up a huge fan of the U, sort of coming full circle and being here right now is just really cool and special for me. Now it's not working. All right, we just do this. But to really get started, I wanted to, of course, share today all of the work I did throughout my PhD, which centered around figuring out how ectoparasites fit into coral reef food webs. I don't know what the heck that is. So the kind of gist of the talk, I'm going to give a bit of background on the motivations for the work I did and sort of where the gaps in knowledge were at the beginning of my PhD. Then I'll sort of go over some of the general collection methods that were present throughout all of my studies, so that way I don't have to go through them again and again and again with every study. And then I'll get into sort of the main research questions that I was trying to answer, and then sort of sum everything up in the conclusion area. I'll talk about some of the future lines of research that my PhD has now sort of set the groundwork for, that I'm really excited about. And then I'm going to thank a bunch of people. So if I talk about trophic structure, food webs, food chains, often one of the first things that people think about is going to be a sort of classical predator-prey interaction, usually involving some kind of charismatic megafauna, right? These interactions they're exciting, they're dynamic, and it's no surprise why that's sort of what we gravitate to more than, say, something that sucks blood that you can't really see. But sometimes these little internal biases that we have towards things that are exciting and really striking seep into the way we ask our questions in science and the things that we focus on the most. And one of those areas has been in trophic ecology. Now, when I talk about trophic ecology or trophic dynamics, I'm talking about the way that nutrients move from the bottom of the food web or food chain to the top. So from autotrophic sort of organisms that make their own food, moving through different trophic levels of herbivores and predators, and basically how the currency of trophic interactions, which is energy, goes from the bottom to the top. And consumptive interactions, like predator-prey interactions, they do impact many other aspects of community ecology. Okay, they can influence behaviors. The top-down pressure of consumption is a major structural factor in trophic dynamics. And one of the main ways that we study trophic dynamics is by creating food webs. So food webs are basically a series of food chains that are overlaid on top of one another to make a more complex diagram. And they're useful for answering questions that often center around what happens if things change. Okay, what happens if environmental variables change? What happens if we completely remove a species? How's the rest of the community affected, and how do things change as a result of certain conditions that might change? And while food webs are an incredibly powerful tool and honestly just pretty cool looking, they haven't come without their criticisms. So a lot of the early food web theory was built on some sort of fundamental assumptions that it turns out are not quite 100% correct, and they don't quite fit everything. And this has sort of resulted in what is an uneven resolution in most food webs. So if we take a look at this food web I pulled up here, it comes straight out of the textbook. If you look at the top of it, you have really fine detail on the upper level species and the charismatic species. You have a leopard seal, you have a sperm whale, very specific species that are finely detailed, that have a lot of quantitative data on their interactions. But as you move down and get progressively smaller, you have very broadly grouped organisms, such as phytoplankton or copepods. And so a lot of these are grouped based off of size and not necessarily how they fit together functionally within the context of the web. So as you get smaller, you lose some of that detail in terms of how these different things might be contributing to the overall structure and function of the community at large. And so if we sort of take that food chain that I isolated earlier and we ask ourselves, where are the parasites in here? And one of those assumptions that I mentioned that a lot of food web theory was built on was a size-based hierarchy of consumption. So that means that things eat things that are smaller than they are. So if you see here, this, again, really simplified food chain, our top predator eats things that are smaller than it, and everything down the line eats things that are smaller than they are. And the fact that parasites break this rule and also are just really cryptic, right? You don't see them very often. A lot of times they're living inside the host, or they're pretty inconspicuous on the outside of the host. That combined with the fact that it was always just assumed that because of their small size, any contribution they made to the structure or energy flow in a food web was negligible at best. And so including them wasn't really important. But if we look at where parasites would fit, what? Oh, okay. If we look at where parasites would fit based on a size-based hierarchy, they would go here in the bottom with the autotrophic organisms that, again, they synthesize their own food. And so that would suggest then that they should be eaten by things that are bigger than they are, and they should not feed on anything bigger than they do, which of course is not the case with parasites. Most parasites can feed across various trophic levels, and they almost exclusively feed on things that are larger than they are. So they break that fundamental rule, one of those fundamental rules that the initial food web theory was built upon. Now, I say that parasites have largely been omitted in terms of food web studies, but that's not to say there hasn't been some really great work done looking at how they impact food webs and trophic structure. So largely starting in '97 with this paper that was titled Food Webs, a Plea for Parasites, there have been an increasing number of studies that show that, in fact, yeah, parasites are major players in the structure and function of food webs, and they're major players in trophic dynamics. And so two of the main types of food webs that we use are, one, topological webs, and two, energetic webs or bioenergetic webs. And now in food web topology, what we're mainly concerned with are what species are present or what functional groups are present. So here the boxes represent either a group of similar species or individual species. And then the lines connecting them represent some kind of a trophic interaction between the groups. I kind of look at topological webs like Jenga. We're not concerned so much about some of the finer details. We're concerned about what species are present and how many things are they connected to. And so if we look at the Jenga blocks on the left, we see there's a lot of different connections and a lot of species within each level. So this system on the left, if you removed one block, the whole thing isn't going to topple over. But the one on the right, is much more susceptible to, if you remove one or two species, sort of weak blocks, such as removing these here, then everything on top of that falls over, right? So I kind of look at topological webs like Jenga. And we know that parasites can impact food web topology. So this food web I showed you earlier was one that they made for a sub-Antarctic lake, a community of organisms in a sub-Antarctic lake. And this one here they made with no parasites. And then they made the same food web with parasites. And so all the red interactions and all the species on the side are parasites and parasite and interactions that involve parasites. And when they added parasites, parasites comprise more than half of the interactions in the entire system. They increased all of these different metrics that we use to evaluate trophic structures. So things like links per species, connectance, which is a derivative of links per species, average trophic level, and average food chain length. These are all things that we use to determine how stable and how robust a system is. Now, energy is, like I said, the currency of trophic interactions. And most organisms, they need to find food, so they get energy, so they're fueled to do all the things that they need to do in their life. To spawn, to maintain their habitat. And while for most organisms on the reef, it's food where they get their energy. For a lot of us in here, it's coffee. And Paul's definitely no exception for that. And we know that parasites can influence energy flow. So there was one study that found in 2008, published in Nature, that found that parasites can comprise more biomass. So not necessarily looking at the transfer of biomass, but they can represent more stuff, more material than top predators. And they're comparable to a lot of other functional groups in this stream system. And then also in that same stream system, they published a study more recently where they found that parasites can in fact transfer more biomass than predators in total from a focal group. So they were looking at snails in this stream system, and they found that when it comes to snails, parasites are in fact moving more biomass than predators. But the stream system is pretty simple. It's not super biodiverse. It's not a very complex food web or food chain. Now, in coral reefs, the most common ectoparasite that we find of fishes specifically are Gnathiid isopods. In the Caribbean, the main species that we have is Gnathia marleyi, named after Bob Marley. You can thank Paul for that one. And so Gnathiids, we refer to them as the ticks of the sea because similar to terrestrial ticks, they latch onto a host and remove a blood meal. Now they live in the benthos, generally in the bottom habitat, and then they will emerge. They'll attach to a fish host. They'll remove a blood meal. Then they'll go back down, digest for a bit, and emerge as a second stage, unfed, a little bit bigger. They'll do it again, feed, go down, molt, come back up, a third stage. Then as a third stage, they feed one more time. And then after the third stage, they go down and they metamorphose to their adult stage. They do not feed as adults. They only feed, they're only parasitic and only feed during their juvenile stages. As adults, the only thing they do is mate and die. And I should have mentioned there, they are most active during crepuscular times, right? So dawn and dusk, and also more active throughout the night. In general, gnathids aren't very active during the day. We know that gnathids can have some real heavy impacts on host fishes. So settlement stage fishes, a single gnathid can kill a settlement stage fish. In juvenile fish, which of course are a little bit bigger, a little more robust, a single gnathid can impact the escape response, so making them more susceptible to predation. And then in adults, the presence of gnathids impacts their spawning behavior, influences their movement behavior, can increase stress hormones, and can lessen the quality of their blood by lowering hematocrit levels. And in high infestations, gnathids can kill an adult fish, can kill like rather large adult fishes in a high enough intensity. Now, on our average coral reef at our sites, well, reef, there's not so much coral these days, but a reef, we of course have our divers who are looking at the fish. We often have our territorial herbivores. We have our benthic invertivores represented by our little blenny here. We have the rovers, the either roving herbivores or detritivores. We have our scrapers, which are represented by the parrotfish. And then, of course, we need to have our cleaners. We have the dedicated cleaners and the facultative cleaners, and I'll break those down in a little bit more detail later on. And then, of course, oh, how can I forget? We need our soft-bodied invertebrates, because what good is a reef without some inverts? Then we need to have some of our predatory fishes that apply that top-down pressure of predation. But then, of course, you need to have a top predator to sort of control the whole system. Now, if we were to look at the classic predator-prey interactions that would be present in this system, it would look something like this. In most food webs, this is what they would focus on, these classic consumptive predator-prey interactions. But when we do that, we're then ignoring all of these Gnathiid-based interactions. And Gnathiids are only one parasite. Okay, there's tons of different kinds of parasites with different consumptive strategies that also can infect a wide array of fishes. But in terms of Gnathiids, we've now confirmed, I think in Gina's paper, we're up to 83 different species that Gnathiids will feed off of. So they are generalists in their infestation, even if they do seem to have preferences for certain species of fish. But to omit parasites is to ignore all of these different trophic links, which, as when I mentioned topology, food web topology, this certainly makes a difference in terms of the topological qualities of the system. And then, of course, we know a little bit about what eats Gnathiids. So we know that coral consumes Gnathiids, live coral, and we know again that cleaners eat Gnathiids. And so cleaners are individuals that remove parasites off of other fishes. So they're kind of like the car wash for fish, where the fish come up, they get their parasites picked off, and they're a little bit happier on the other side. But so we know that Gnathiids feed on a lot of different fish species. We know a couple things that eat them. Where are the gaps in knowledge here to sort of get a better idea of how exactly they contribute to community level trophic structure? And that leads to my research questions, which there were 4 questions that are then sort of broken out of 3 dissertation chapters. The first of which is, do Gnathiids only feed on fish? The second is there a Gnathiid free zone, which means, is there a place where a fish could perhaps go to avoid infestation altogether? What non-cleaner fish species consume Gnathiids? And the final one, which gets into the energetic side of food webs, is how do parasites, specifically Gnathiids, contribute to biomass transfer? So before I jump into those questions, I want to introduce sort of where we did these studies and also some of the collection methods we use. So all of the studies that I completed during my dissertation took place in the Caribbean. We sampled in La Parguera in the southwest of Puerto Rico. We worked on Isla Culebra, just off the coast of Puerto Rico, in the US Virgin Islands on St. Thomas and St. John, and in the British Virgin Islands on Guana Island. Now, not all of the studies took place in each site, but collectively, those are all the sites involved in my dissertation. Now, how do we collect Gnathiids? There are two methods that we use to collect Gnathiids, the first of which is via light trap. Light traps are PVC tubes with a funnel on the bottom and a filter on the cap on the top, and there's a flashlight that shines out the bottom. We'll put these things out usually towards the end of the day, and then as it gets dark out, the lights attract a wide array of plankton species. We pick them up the next morning and then filter them and pick out all of the Gnathiids out of the plankton soup. The other way that we sample for Gnathiids is with cage fish. So we will catch fish, put them in a small mesh cage, put them on the reef for a set amount of time, recover them, put them in a bucket, and then the Gnathiids, after they finish feeding in a couple hours, will drop off naturally. How do we catch fish? Because I'll tell you right now, we don't go fishing with a hook and reel like most people do. The way we catch fish is on snorkel or scuba. We use a wide array of nets. We use sometimes barrier nets that are weighted on the bottom and have floats on the top, so they sort of make a wall that we can chase fish into. We'll use, like I'm using here, I'm using a cast net that we modify to remove the pulley bit. So it's essentially just a net that's weighted all the way around on the outside. And the way that we catch fish depends on what fish we're going for. So all the different species on the reef sort of react differently to being chased, or they sort of have different tendencies. So we have to adapt our method of catching based on who we're trying to catch. Now getting into my first research question, do gnathids only feed on fish? So we've always referred to gnathids as fish parasitic. We know if we put a fish on the reef, they're going to come back generally with lots of gnathids. So we've always just assumed that maybe they're obligate fish parasites. But then as we sorted through a lot of our light trap samples, we found repeatedly that gnathids we find our gnathids attached to soft-bodied inverts. So here's a gnathed attached to a polychaete worm. And we kind of wondered, what's making this happen? Because light trap environments can be kind of mixy, right? There's a lot of different chemical cues going on. There's a lot of plankton in a small place. There's often a lot of fish activity on the outside because they get pretty excited with all the plankton. So, there's a number of things that might result in them attaching to an invert. So for this research question, we wanted to figure out, will gnathids feed on, in an isolated setting, soft-bodied inverts, and could they feed on them, and could they metamorphose from a meal that they got from an invert? And so in St. Thomas, where we did this, we collected some of the most common soft-bodied inverts around. We collected the lettuce sea slug, the bearded fireworm, the spotted sea hare, and the octopus, the Caribbean octopus. And so we put them in their own containers. We threw some wild-caught gnathids in with them, and we let them sit overnight. And then we looked the next morning to see if anybody fed. And it turns out, yeah, in fact, gnathids will feed on soft-bodied inverts. So we had individuals that successfully fed on the bearded fireworm and the lettuce sea slug. The one individual that fed on the bearded fireworm died, but of the 9 that fed on the lettuce sea slug, 5 molted to male. This is one of the males from that group. 2 to female, and 1 molted to the next juvenile stage. So out of the 9, 8 of them molted successfully off of a fluid meal that they got from an invertebrate, something that we never really would have thought possible before this. And so what does that mean? That means that now we have an entirely new group of organisms to consider as potential hosts. So yeah, we've established a new feeding link, which goes towards the topological qualities of most Caribbean food webs. But now we have to think, under what circumstances on the reef in nature might a gnathed feed on a soft-bodied invert instead of a fish? Will they ever choose a soft-bodied invert over a fish? So there are definitely some lingering questions there. I mean, maybe if a gnathed ends up in an area where there are the less preferable hosts, like parrotfishes, they don't really like parrotfish very much, it seems. Will they maybe feed on a soft-body invert to sort of hold themselves over until maybe there are some more preferable fish species, or maybe they get trafficked off on a host somewhere else to end up in a better area with more available hosts. Now, moving into my second question, is there a, quote, gnathed free zone? So previously, there was a study where they estimated the distance along the benthos, the distance along the bottom parallel with the bottom that a gnathed might travel to attach to a fish host, to attach to a caged fish. They estimated roughly a meter, that most gnathids were probably traveling about a meter to get to their host. But we had never considered how gnathids might use the vertical space in the water column to exploit hosts, to infest hosts. If we think of their terrestrial analogs, ticks can't fly. So there's no real vertical component to their infestation, but gnathids can swim. So there's potentially a completely different array of fishes that spend most of their time above the benthos that may or may not be very susceptible or see very high infestation rates, and it's dependent on how high will a gnathed go. And so that, of course, was the overall aim of this question, is to find out what's the vertical limit of gnathed infestation. And so to do that, we suspended fish in the water column. And this study, I remember very well. We were in St. Thomas, and I had not conceived of this study yet. And we had put out some marker buoys to mark some of the areas where we were light trapping. And the marker buoys were similar to these lines, where they had floats at the top, so we could find the area, and then they were tied to the bottom. And I noticed over the period of a couple months, as we had these lines out, the growth along the line, as you move deeper and deeper, got less and less. And so I saw this just decreasing amount of growth as you went down. And I said, I wonder if gnathids differ in how they sort of exploit the vertical space. That was the moment, I'll never forget it, where I sort of thought, like, maybe we can just suspend fish in the water column and see if any get gnathids. And so that's what we did. We did this in La Parguera, and we did this in St. Thomas. And we, of course, also set individuals on the bottom, since we know we get gnathids on the bottom. So it served as a good comparison to compare with the guys in the water column to see what our infestation levels were like. And what we found was that there was really no difference between 0 and 1 meter. They got roughly the same amount of gnathids. But then as you went from 1 to 2, 2 to 3, 3 to 4, you saw decreasing amounts of gnathids as you went higher. The highest height that we found in gnathiid, though, was 3.5 meters above the surface, which compared to the roughly 1 meter they were estimating for what I'm calling horizontal movement or movement parallel to the benthos, that's a much longer distance. And it's way farther than, honestly, when Paul and I talked about it, we thought they were going to go. I thought that this curve was going to be much steeper. So yeah, gnathiid vertical movement is way more than we would have considered. And this makes it more difficult for hosts to avoid getting hit by gnathids. And it potentially opens up this this trade-off where if you're going to move to the position where you are, if you're a host fish, potential host fish, and you want to move to the position where you're going to miss out on most of the gnathids, you're going to find yourself 2, maybe 3 meters above the benthos, which then for a lot of fishes is going to make you more susceptible to predation, because you lose out on all the habitat that you can then dip into and use to avoid predators. And it also suggests that maybe the gnathids that are higher up in the water column are avoiding certain micropredators. So certain fishes like squirrelfish are nocturnally active, and we know that they consume gnathids at night. So the guys that move up into the water column are maybe avoiding some of those nocturnal predators that might eat them. And then this sort of opens up gnathids as potential vectors of dispersal, right? So gnathids don't have a planktonic phase, and they don't really swim long distances. They have really good acceleration to get to a host, but they're not going to swim very far. So some of the fishes that spend most of their time higher up in the water column, like some scombrids, some of the jacks that we see, they may serve as sort of vectors of dispersal that can move and transport gnathids to adjacent habitats. All right, now moving on to question 3. What non-cleaner fish species consume gnathids? Now here's all of our interactions again, and then we have been sort of thinking about the gnathiid-based interactions that gnathids feed on a lot of fish, and we know that again cleaner fish eat gnathids. And in fact, this interaction with cleaner fish, cleaning host fish, and removing gnathids is what got most of the gnathiid scientists into studying them. It was these interactions that made people like Paul say, huh, what's driving that? And so most of the people that study gnathids got into it through this exact interaction right here. But okay, cleaners eat gnathids. There's so many other fish on the reef, many of which consume invertebrates. So what about the other direction of this interaction? What other fish species might be eating gnathids? And so that's where, of course, the aim of this question comes from. When we're thinking of the stability of a system, functional redundancy is something that is important. Okay, if you have a role in the reef that has low functional redundancy, then those individuals that fill that role, if you lose them, you potentially lose out on that service or that role altogether. The higher the functional redundancy is, meaning the more individuals that fill that role, the more you're able to absorb the loss of one species or one group. So what eats gnathids? Again, dedicated cleaners. In the Caribbean, it's gobies. Gobies are very tightly associated with live boulder coral. They eat gnathids exclusively. We have cleaner shrimp that will remove gnathids from fish. And we have the facultative cleaner, the bluehead wrasse is a primary facultative cleaner. Facultative cleaners, they eat gnathids, they clean sometimes, either only part-time or only during certain life history stages. So for bluehead wrasse, it was always thought that as juveniles, they're more likely to clean, but as they get older, as they get bigger, they become less likely to clean, or they just stop, altogether. And again, gobies are very tightly associated with boulder coral. So we see here, there are 3 different gobies on 3 different types of corals. And the fish that solicit their cleaning interactions will pose oftentimes vertically, like you see here. They'll swim up and sort of posture their body in this pose, saying like, hey, I've got some parasites on me. I need some help here. Or they'll just come and rest on the coral head itself, and then the goby hops on and goes to work. Okay, but what else eats gnathids? Like I said, nocturnal microcarnivores, squirrelfish, French grunts, they eat gnathids, but we see they don't eat anywhere near the number that cleaning gobies do, right? So much lower for everything else. We also know, again, live coral. Live coral eats gnathids. But again, what about all the other fish? This little rosy blenny, benthic invertebratus, eats little inverts off the bottom. Thought maybe. So to collect our samples, we caught fish like I described before using a variety of methods, and I took their stomach contents either by lavage, which is a stomach pump, dissection following after a humane euthanasia, of course, and when the lab facilities were sufficient, we would keep the fish. I would collect their poop, and then the fish got to go home, and then I would just look through their poop. Holiday conversations with my family are really, really interesting. So in total, across Puerto Rico, St. John, and St. Thomas, we sampled 716 individuals, and we found, unsurprisingly, all of the dedicated cleaner gobies consume gnathids. Found that 64% of the part-time cleaners, we'll call them, consume gnathids, and then everything else under that was pretty low. You might say, oh, hey, but the planktivores, 33%, that's pretty decent. They're doing okay. But if we look at the numbers that they were consuming, even though 33% of the planktivores, yeah, they ate gnathids, but when they ate a gnathid, they typically only ate one. And so this plot on the right is only counting individuals that did eat gnathids. And this one on the left is including everybody, whether you ate one or not. So we can see that when you eat one, if you're not a cleaner, you really only eat maybe one or two. You're really not eating very many. But the cleaners, dedicated and facultative alike, they went to town. And there, in fact, was no difference statistically between these two groups, which honestly, even though we expected a lot of gnathid consumption from facultative cleaners, I did expect the dedicated cleaners to beat them out. And also in terms of the facultative cleaners, I found gnathids, tons of gnathids in juveniles, adults, in males, which I certainly didn't expect. So they seem to be doing a lot more work than we would have anticipated. And so another way to look at this is looking at, I calculated, okay, based on all these different consumption rates, how many individuals would it take from a given functional group to replace one dedicated cleaner goby? One fish that's about this big. How many individuals from each group would it take to replace one? And for these three groups, it would take at least 150 individuals, 150 roving carnivores to replace one dedicated cleaner. 150 of all those groups. The rest of these groups here, at least 100 benthic invertebrates just to replace one little goby. The only thing that was a one-to-one or close to one-to-one substitution were the facultative cleaners. So now we have found out, we've discovered now that these interactions, which I was previously showing as a one-way interaction, these are all two-way interactions. These are brand new trophic interactions, and now gnathids are big players, not only as consumers, right? They don't just feed on a lot of fish, but they get fed on by pretty much all those same species of fish. And so now our web it's growing a bit, right? If we started out with only the red marks, and now we have all these different gnathed-based green arrows that are really filling out this web. And now, this picture is looking a little different than it did before. And so in this question, in this study, we have described several new trophic links that we didn't know really existed before. But the fact that they're weak interactions suggests that, in fact, the functional redundancy of gnathed consumption is pretty low. And so these cleaning mutualisms, these interactions are now perhaps more important, or now they perhaps look a little more important than we thought before, because if you lose your cleaners, there's nobody that's really going to pick up the slack. This also makes more species susceptible to transmission of bloodborne parasites. We know that gnathids will remove bloodborne parasites from hosts and then keep them in their gut. And so then if a certain fish consumes that gnathiid, they then potentially assume and become infected by those bloodborne parasites. So now all of these fishes that we've described these feeding links for are now potentially vectors for bloodborne parasite transmission, or the sort of terminal host for it. And this also opens up the idea, based on this and based on the fact that we know, again, they feed on a lot of different fish species, are gnathiids maybe the most trophically connected group in the ocean. I can't think of anything else that is fed on by as many organisms and feeds on as many organisms. Final question now. How do they contribute to biomass transfer? So we know that they touch a lot of stuff, right? They're interacting with a lot of stuff. But what does it look like in terms of the currency? How are they functioning as bankers of biomass? And this, again, this looks at them as consumers, as predators. But if we look at all these interactions here, to quantify the biomass transfer of all of this would have been a little bit out of the scope of my PhD. Would have taken a little too long, I think. So what we decided to do was isolate certain interactions, quantify them, and compare them to one another. Specifically, we wanted to focus on Gnathiids damselfishes and quantify how much biomass is being transferred by predators, how much is being transferred by gnathids, and how do they stack up to one another. We chose damselfish, the Gnathiids damselfish. Number one, they're really easy to catch. They're territorial, so they're very easy to monitor, and they're present at all of our sites. Now, to determine biomass transfer by a gnathiid infestation, we caught a bunch of fish, individually housed them in buckets. We had all kinds of air stone lines going crazy, taped on the wall. We then took a set of 10 to 15 fish, caged them, put them down on the reef during each of these time blocks for 45 minutes that were equally dispersed throughout a diel 24-hour cycle. We then recovered the cages, put them in their own buckets, let the gnathiids feed and fall off, and then counted and staged all the gnathiids that we collected. We also did this during some of the peak times, sunset, sunrise. We did some comparative sets where we set out adults, intermediates, and juveniles to look at how infestation changes as you get older, if you're a damselfish. And this is some of the damage that I did to people when we were sampling at midnight or 3 a.m. or 6 a.m. This was, I think, 3 o'clock in the morning. We had fish in the water. We were waiting, and everybody's using life jackets as blankets and napping. This particular day, I'll never forget. This was in Guana, and we had just finished a sampling set, a series of sets that began at 7 p.m. and we ended at 10 a.m. We went literally throughout the entire night without a break. We had multiple groups of fish that were like, one group goes out, they come back, the other one goes out, they come back. These guys are sitting and stewing, and it's just boom, boom, boom. And yeah, everybody was absolutely blasted after that night. And we did this, these 24-hour sets at 5 different sites. So I abused a lot of people. Now, to quantify biomass transfer, we caught and tagged damselfish. Adults we tagged with external Floyd tags, and the juveniles we used a subcutaneous elastomer tag. I have a little bit of the elastomer in my hand still. If anyone has a black light, I can show you how it glows. And so we monitored these guys for about 4 weeks, looking for signs of predation. So because they're territorial, we felt pretty confident that if they're not in their territory or they're not nearby, they're probably been eaten. The intermediates and juveniles, though, we only monitored for 2 weeks because, for reasons you'll see, that was pretty much all we needed. Now, the patterns of diel gnathiid infestation, so 24-hour infestation, this sort of relationship is very similar to what Paul found in 2006. You have a peak pre-dawn right before sunrise. You have a peak post-dusk right after sunset. You have a bit more activity throughout nighttime, and daytime's really pretty dead. We also, to sort of cover all the bases, sampled for some additional parasites. We did freshwater dunks for other permanent ectoparasites, And we also, I dissected certain individuals to look for endoparasites. So ectoparasitic copepods and maybe some nematodes. I'll tell you, this did not come from a damselfish. This is from a different fish I dissected. And I'm going to put this table up, even though I'm not going to explain it totally. So you're going to have to hear me now and believe me later on this table. But I wanted to point out that for all of the other parasites that we sampled, there was just really low prevalence, really low intensity. There was not much. Damselfish really were not getting hit by much else besides gnathids. So for the rest of the biomass calculations, we decided to omit them because there just wasn't very much going on. And I didn't want to go into great detail about it, but I wanted to mention it briefly. Now for the life history comparison sets, we found 23% of juveniles got hit by at least one gnathiid, 52% of intermediates, and 82% of adults. And when I modeled this out, we had significant effects of fish length and of site. So we did have differences across sites, which is interesting, but catching gnathids can be kind of hit or miss. So I would say that, for instance, in St. John, even though we didn't get very many here, I would not say that's indicative of the population overall there, because in other sets in previous times, we've gotten absolute huge loads of gnathids. But what we can see is that right around 5 centimeters, which is when a lot of the Stegastes start to show their full adult colors, we see sharp increases for most sites, pretty big increases in predicted gnathiid infestation. And for those individuals smaller, there's really not much going on. Now, the results for the predation, the consumption, the green represents the adults across sites. We saw really high survivorship. And in fact, La Parguera, none of the individuals were consumed. Guana saw the highest consumption at, I think, 23%. The juveniles really did not fare very well here, and the intermediates were intermediate. And so looking at this in terms of biomass, we see that as juveniles, there's a bit of predation-based biomass. So X here is predation biomass, Y is parasite-driven biomass. We see there's a bit of predation biomass, not very much parasite biomass. Transfer, right? As we move to the intermediate stage, we see a pretty decent little jump, more than double in predation biomass transfer, but not much of an increase at all in parasite biomass transfer. But then as we move to adulthood, we see a big jump in predation-based biomass transfer and a big jump in parasite biomass transfer. So with predation, we see a pretty consistent increase as you get older, and parasites not doing very much for younger stages, but doing a lot more for adults. Now, the same X and Y axis with parasite on the Y and predation on the X, we look at the site-based differences for adults. We see that here, La Parguera saw the highest parasite biomass transfer, estimated parasite biomass transfer, and Guana, in fact, was second. And Guana was also the highest predation biomass transfer. But we also see that across sites, the parasite-driven biomass transfer for both of these sites was higher than all three of these sites, and of course, Parguera as well. So these two were higher than 4 of the predation-driven biomass transfer. And then looking at transfer relative to what's available, I wanted to point out, number one, that juveniles and intermediates are not making up a very big proportion of the overall biomass. As damselfish hit adulthood, they really fill out in terms of their overall mass. And then I also wanted to point out here that in Parguera, we saw an estimated 0.83 grams per meter squared. So this is per unit area transferred per month. And this represents about 12% of the overall available biomass. Now, compared to the other sites, this 12% of the available biomass being transferred was higher than all 3 of these sites. So on a per unit area basis, relative to what's available, parasites are kind of competitive with predation in terms of how much material they're moving. And so, yeah, for damselfish, Gnathiid-driven biomass transfer is it's a player. It's not clearly beating predation, but it's competitive. It's worth including in studies and in concepts that are looking at biomass transfer. So whereas before we would have assumed pretty much all of the consumption-driven biomass transfer is going through this vector right here, but in fact, this vector, which is part of a completely different food chain, a completely different chain, potentially a longer food chain, and also potentially going in several different directions based on who's eating them. This is now something that we have to include, we have to consider. We can't just omit it, assuming that it's a drop in the bucket. And so overall, predation represents, even if it is, if it's more than parasitism, it represents a smaller proportion of the transfer that's going on in the system. Now to sort of bring everything home here and put a bow on it, we have, through the course of my dissertation, we've number one, established a new trophic link to a brand new group of organisms in invertebrates. Of course, the second part of that, I didn't have a good image for this, but we've now gotten a better idea of their sort of spatial range that they operate inside of. We've also established all of these new links featuring gnathids as prey items, which have also served to underpin how critical cleaning interactions are. And we've now quantified exactly how much material is going out from a focal fish genus to gnathids and to predators. And so moving to a bit of a bigger picture, in coral reefs, ideally, we have lots of live coral. Now, I know these are branching corals. They should be boulder corals, but I didn't have a cartoon of a boulder coral. So pretend they're boulder corals. When we have lots of live coral, we have lots of suitable habitat for gobies, because like I said, they are tightly linked to coral. They can live on, say, sponge, but they clean way less often if they're on a sponge. So we have gnathiid consumption through corals. If we have suitable habitat for gobies, which is good for gobies, and all of that together is bad for gnathids. But as we see things like climate change come through and leave a wreckage like this, we have less suitable habitat for cleaner gobies, also more suitable habitat for gnathids, because gnathids love them some dead coral. So that is bad for gobies and good for gnathids. When this is the situation, bad for gobies, good for gnathids, we have fish that are not very happy. A lot of our fish will probably see increased parasite burden, which then could have individual community or individual population and community level effects. But what about those cleaner individuals, such as the facultative cleaner bluehead wrasse, that they're not tied to coral necessarily? They're not necessarily tied to a specific habitat structure. So in the absence of cleaner gobies, say, do some facultative cleaners upregulate their activity as a cleaner and potentially service more clients, because it's generally thought that they service a narrow range of clients compared to gobies? Do they change in response to changing goby populations? I don't know, but that leads to one of my main future directions, is looking at what makes facultative cleaners clean versus eating other stuff. What drives the differences across apparently some individuals? Is it population based? If you have more conspecifics around, are you perhaps more or less likely to engage in cleaning? Yeah, so what's driving that? I don't know, but I'm very curious about it. And also, I have to mention that I spent a good bit of time in Australia as part of my dissertation. Initially, the chapter which looked at what fishes are consuming gnathids was meant to have a comparison with Pacific species from Australia. But it turned out that after 3 trips and over 500 fish samples from here, it wasn't near enough to really characterize the biodiversity of this area. So I didn't include it here, and I intend on finishing it after with a few more trips to lizard, hopefully. But it just, the biodiversity there compared to the Caribbean is just insane, way more than me, who had never been there before, really would have imagined. And so even with a good amount of samples, it just wasn't enough. And also, sorry, this table's not great. This is straight out of one of Paul's papers, the 2013 paper. But this was a comparative study looking at the relative susceptibility of different fish species. So in my dissertation, I quantified how much biomass is being removed by gnathids from Stegastes. Well, it turns out Stegastes are in fact one of the least susceptible fishes to gnathids. They don't really get hit compared to other fish species. They don't really get hit that bad. Yet biomass is the biomass transfer from gnathids is still pretty significant. So what about some other groups? What about some of the larger predators that aren't getting eaten by very many things, but certainly get hit by gnathids? And if we even look at the grouper on the wall here that's in the middle of getting cleaned in that picture, what about a huge grouper like that? How many parasites, how many gnathids could that guy have and not even feel it? I almost brought the big grouper that's hanging on the 3rd floor of Grosvenor, but it was a bit too big to bring in. And then what about gnathiid biomass being trafficked out? So some of these more transient mobile groups of fishes that are more than likely getting hit by tons of gnathids and maybe shipping them off to adjacent habitats. That's another aspect that I'm interested in, is looking at the connectivity among adjacent habitats via mobile individuals. So now I move into the part that makes your throat tighten up a little bit. I would first like to acknowledge all of the beautiful sites that I got to experience during my PhD. This is only 2 of them. This is Maho Bay in St. John prior to the hurricane, and this is Trunk Bay in St. John. But there, I could have filled this whole thing with images of pretty vistas. I was super fortunate to see some incredible places and work in just some breathtaking areas. The various labs and facilities that we worked at, sometimes they were a bar or a house. All of the different places I got to go and work, Lizard Island, Ann and Lyle there are incredible. University of Puerto Rico at Magalhães, the whale lab was super helpful. The DSO there, Milton was incredible. Veers, which is I believe still no longer functional, but man, we had some incredible times working at Veers. Guana Island, which is again a private island with, we had to stay at a resort there. The UVI, University of the Virgin Islands, I spent so much time on St. Thomas, and yeah, this work certainly would not have been possible without all of these places helping to facilitate it. Arkansas State, where I started my PhD, specifically, I'd like to shout out Jerry Ferris, who was part of my committee while I was there, and Tom Risch, who really helped facilitate our work. And both of them were really in our corner when we wanted to just travel all over the place all the time. Everybody here at Rosenstiel School, since we've joined up, and all of the work that everybody did to make it a seamless transition for us, I could not be more thankful. I could not be more happy to be a hurricane. Funding, NSF, man, we got a lot of money from NSF, and I think we spent it well. AMLC also awarded me a small grant that I was super appreciative of. My Army, holy crap, I worked with a lot of different people. Every year it was a different swath of people for the most part. This work was 100%, never would have been possible without every single person that I ever came across during my PhD. If you ever caught a fish, sorted through a trap, or just made a cup of coffee, man, I am in your debt forever. Far too many people for me to thank individually, but yeah, wow, I could not have done this with the help of so many incredible people. My committee members, Mike and Martin, you guys are great. Thank you so much for coming on. I wanted to find not your headshot pictures, thank you both so much for coming on and just sort of jumping right in and just making everything feel seamless and fun. And then of course, Marilyn and Dave, thank you for sticking with me through ups and downs and not just saying, hey, I'm done with you, dude. You guys are honestly all really inspirational. I appreciate all of you, and you're all just pretty cool, to be honest. So I appreciate all 4 of you very, very much. Sikkel, holy crap. I'm supposed to thank this guy, but if I'm being honest, He didn't really do much. If you look at all these pictures, he's always out in the sun. He's taking a bath. He's covered in tie-dye. He's holding a syringe here that has a cap on. He wasn't even taking blood from a fish. You know, this is the typical Paul pose. Like, hey, what are you doing there? You know, like. But no, Sikkel, man, you're my guy, Paul. I have loved working with you. I have appreciated how much you have kicked me in the ass when I needed it, but also just let me go and do my thing. You know, you've let me largely go where I want, when I want, and drive my own bus. And but you weren't afraid to really, you know, yank on me when I needed it. And I really appreciate everything. Now I get emotional, man. My core four. Brill, Elizabeth, the silver fox. She introduced me to Paul's work. This picture is from 2012, which was actually the first time I worked on Paul's stuff. I literally would not be here without you. Court, my best friend. She took up work to drive down here today. Andres, I don't know if you made it in Zoom, but Andres was also doing his PhD, you know, alongside me. This guy was my right-hand man. We had so many good times together. We got so much done together. He's just my guy, man. I love him. He is my brother. And Gina, you're my partner in crime, man. I hate you as much as, you know, you're great, but yeah, all four of you are absolutely the four pillars of my dissertation. And I would not be up here if it wasn't for all four of you. I love you all deeply. And my family. Josh, you're my duo. My dad, who's not here right now, he was in the middle of getting a procedure, not serious, but you're why I'm so damn stubborn and why I refuse to accept anything except greatness from myself. And my mom, who my work is dedicated to. You're where my sense of exploration comes from. In the face of some of the greatest adversity a person can go through, you handled it with such ease and just such grace in some of the most difficult things a person can go through that you've been through. And you are my inspiration every day. And so that comes to the end. I always like to throw up this picture of Andres. We were in the middle of sampling and he decided to mess around and pretend to be a dolphin. And it's always my reminder that I am so lucky to do what I do, and science at the end of the day is fun, and we should enjoy what we do. And with that, I would like to borrow a line from one of my favorite movies and ask, you got any better suggestions? And I would love to take any questions. Thank you. Sorry if I went a little long. I don't know when the timer started. All right, that was fun. Yes. Lynn, sorry.
Yeah, it was really very interesting. I never heard a dissertation defense like this. It's a very intriguing subject. I am particularly intrigued by the idea of gnathiid transfer in the guts of soon-to-be prey fish.
Okay, yeah.
What do you know about that? Is it, I mean, don't they need to breathe, or they can hang out in the digestive juices or whatever?
So the thing is they do break down, but because of their sort of hard exoskeleton, they tend not to break down all the way. You can find remnants of them even like very deep into the digestive process, and specifically it's the heads. They just have very characteristically shaped heads, and they really stay intact very well throughout digestion, such that you can even like identify them in fecal matter. So they have been completely through the system and come out the other end, and you can still tell, and sometimes you'll find the whole kind of husk of a body, and it looks to me like, I always say it's a ghost Gnathiid. But yeah, just the hard parts tend to come through digestion.
Okay, so we're not then talking about the idea that they could prey upon the predator of the fish who's gut buried.
No, not necessarily. I think once they get consumed, they're done for.
Okay.
But in terms of connectivity, it's more the idea of, are you hitching a ride on a host, and then they're sort of shipping you off to wherever they end up, when you fall off.
Okay, great. In terms of energy transfer, but not.
Yeah.
Not a viable way that they can find.
Right. No, I'd be very surprised.
Okay.
Yeah. For the people on Zoom, can you repeat the question?
Yes, I will. Sorry. Yes.
It's interesting that your different sites had such different characteristics and things that make them speak and all that. Did the sites vary in things like fish biomass and coral cover?
So, it's something that really I thought a lot about, and obviously there are some really stark differences. Overall, our sites are very low coral. There's maybe some variance in coral cover, but it's pretty similar. Fish biomass, pretty similar. The thing is Gnathiids are really patchy. So even if you have really suitable habitat, say a bunch of dead coral, you can find a habitat that ticks all the boxes, but you put a cage there sometimes and you don't really get much. And then you put a cage a meter away in the same exact everything, and you'll get a huge load. So the way I look at the differences across sites is they're more just representative of how patchy Gnathiids are, and not necessarily indicative of differences in populations across sites, but more just representative of what Gnathiid populations can be like in the Caribbean, really. Yes, Lynn again, and then I'll go back to you, sorry.
I'm also wondering, you say, we just talked about patchiness, so I'm sure you don't have anything but a feeling about this.
Mhm.
With the decline of stone corals and thus the likely habitat for cleaners, are fish dirtier? Are they more effective?
So in a study that we published in 2020, we did find that there were more Gnathiids in areas with less live coral. So overall, I would say if you're in an area with low coral, like most of our sites, I think the fish are going to be just a little grungier than if you have a high coral site. I'll say it's not a good direct comparison, but I dove on a reef in Australia with the highest coral cover I've ever seen in my entire life. And the fish just looked like they were glowing, to be honest. And there's a lot of cleaning activity there too. And the fish it's anecdotal, right? And I was also just, amazed by all the coral I saw. So there may have been a bit of that, but they did just look robust and hefty, where sometimes on some of the reefs that we know we get lots of Gnathiids, some of the fish look a little grungy. Yeah. Yes. Thank you. Thank you.
I was really interested in the vertical range study.
Hmm.
So I'm wondering if you think that the fact that it's light from new meters. So in other words, if they have to traverse both the vertical and the horizontal.
Sure. Yeah.
oftentimes it favors the one each meter.
Yeah, so honestly, it's a great point. It's something that we thought about. We've thought about doing sets with fish in larger cages so they could move around more easily. But that definitely is an artifact that we have to consider, where the fish can't escape. They're stuck in the cage. So I still found it interesting that the Gnathiid would travel that far, period. But you're totally right that a single Gnathiid, or I think on the fish at 3.5, we got like 3 or 4 maybe. I'd have to look back. But it is most probable, I would say, that fish would be able to avoid those Gnathiids if it wanted to, just by swimming out of the way, perhaps. But it is definitely a point that is important. Yeah. Derek. So we have, I think, Gina, what? Sorry, God, I forget to repeat. So the question was, did we find Gnathiids on like some of the more the species that spend more time in the water column, like barjacks or mackerel. I think in Gina's study that's now in review, you got sequences of blood meals from jacks, right? Yeah, so we have found evidence that, in fact, yeah, that gnathids do feed on those fish species. We didn't quite know how that might happen. We now have a bit of a better idea, knowing that gnathids can travel up in the water column. But we also do see some of the barjack getting cleaned by bluehead wrasse. Now that could be other ectoparasites, could be copepods, could be monogeneans, but it also now is maybe more likely to be gnathids for part of that. Thank you, dude. Anywhere else? Anything? Cool. Anyone in chat? I don't think so. Cool. Okay. Thank you. Take stuff on your way out. Thank you, Rachel. Thank you, everybody in Zoom. Thank you very much for showing up. I appreciate you all. I love you all. The whole time I was like, I'm not gonna get emotional. No way, man, not me.
Says, great job.
Thank you, Kobe. I see you. Nightman cometh. I see you guys. Thank you. Sweep. Cheers. Okay. Yeah. Yeah.